Hello everyone, this is a quick video on nitric oxide measurement by DAR 4M-FM dial. This is a rhodoamine-based photostable nitric oxide fluorescent indicator. This has a molecular weight around 430 moles per gram, and this is soluble in the MSO. So this is what happens in the presence of nitric oxide in the cells. In the presence of esterases and oxygen, this reacts and form this DAR4M product, and in the presence of nitric oxide, this forms into a DAR4MT. This is a red fluorescent product, and this is what you can do. There are different methods described by different scientists. So from the stock solution, you can take around 5 to 15 micromolar concentration in potassium phosphate buffer and incubate these plants around 10 to 60 minutes, and it depends on the conditions you use. And after washing with the same buffer, you can visualize these nitric oxide production using a fluorescence microscope around 575 nanometer emission wavelength. And this is how you can visualize under fluorescence microscopy. You can see under stress conditions such as soil stress, there is high nitric oxide production in these Arabidopsis roots, and using image J, you can quantify this nitric oxide production. And good luck.